Okay, welcome to the third and final video in this microscopy seminar series that I'm conducting for the usage of microscopy in biochemistry and biology in general. And today's video is going to cover how do we generate quantitative data using a digital image. So really the question comes down to, how do I turn a picture into a number? And we can do that because the way these images are generated in a quantitative dependent manner. So first thing for turning a picture into a number is we need to remember the light path in case it's been a little bit since you've seen the first video. So some light source is going to pass through the microscope. The objective lens is going to focus the light on the sample. The sample is then going to absorb a photon and emit that photon at a certain wavelength. The confocal head is going to get rid of the out of focus light that is not part of our sample that we care about. The in focus light is then going to pass through the confocal head and reach the camera. The camera is then going to convert the photon into an electrical current, which is sent to a computer, and then the computer is going to interpret these signals and make a pretty picture. So we haven't talked about the camera yet. We know that it's a detector of photons, but we need to talk about how the mechanism of how a camera actually works. Like I say in every video, microscopy is governed by physics and applied by biologists. So this is going to get a little bit technical and a little bit physicsy. I don't want you to get lost in the physics. I just want you to understand what it means and how it works. So the first thing we need to do is we need to define what a pixel is. We hear the term a lot in just your general cameras, right? You have a very pixelated image, means that you can see a bunch of little squares. But what is a pixel? So a pixel is the smallest unit of information in a digital image. That is not very helpful, and it's not very specific. It just means that something is small, right? So the smallest unit of information can be further defined into the smallest photosensitive unit. What is the smallest photosensitive unit? Well, that comes down to how we detect photons. So the smallest photosensitive unit can be defined as the smallest photosensitive component in the camera, and that component is called the photodiode. The photodiode is what's going to actually convert a photon into an electrical signal that can be interpreted by the computer. So in a camera-based system, pixel sizes are determined by the distribution of these photosensitive diodes and the actual physical size of the photodiode. So in the cameras that we use, the photodiode's physical size is 6.5 microns in diameter and 6.5 microns tall and wide, right? So you have a 6.5 micron square that makes up the photodiode. So our pixel size cannot be smaller than 6.5. It cannot be bigger than 6.5. 6.5 is the size of our pixel because that is the size of the photodiode. The number of pixels, which is what I meant right in that last sentence, it's a poorly written sentence, is determined by the distribution of the photodiodes on the chip. So the cameras that we use are about 2,400 photo-pixels wide and by 2,400 pixels tall. So that means that there's 2,400 photodiodes distributed along the chip of the actual camera. So when it's all said and done, the size of your pixel is determined by the size of your photodiode that is distributed on the chip. That's the easiest thing to remember. So modern microscope cameras do not look like a camera that you probably think of when you think of a camera, right? They don't have a lens, they don't have a lot of the other parts of it. The lens of this camera is the microscope. The rest of it works just like your phone or any digital camera you've seen before. It's just using the microscope as the lens as opposed to having a lens attached directly to the chip. And so on this camera, you can see we have an opening. This is what's going to allow light to come through. And behind that opening, there's going to be a chip that's going to detect things. This camera is a Orca Fusion made by Hamamatsu, which is one of the cameras that we use on our microscopes here at the University of Wyoming. So behind this opening, we have a chip. So if we go over here to this cutout, this is an image that is representing what the chip's architecture is. There's a lot of this here that you don't need to care about. That is a computer engineering thing that we don't need to understand. You have sensors and analog signal processors, and all this stuff is what's designed. This part is all designed to talk to a computer. We don't particularly care about that. We care that it talks to the computer and we're done, right? But this kind of brownish tan layer, this layer we do care about. So this is the distribution of those photosensitive diodes I was talking about. So if we blow this up into a single spot, what you can see is we have an array of photosensitive diodes distributed across the chip. So if we're talking about this camera in particular, each one of these photosensitive diodes is going to be 6.5 microns in diameter and 6.5 microns tall. And so then we have 2,000 to 2,400 of these things distributed across this chip. So we're going to have a pixel size of 6.5 microns, and we're going to have an image size that is 2,400 pixels by 2,400 pixels. Those are not changeable on a camera-based system. Those are governed by chip architecture. There are other types of imaging, which I don't want to convolute things too much, that do not use a camera and a chip-based system where you can modulate the size of the pixel. But for our purposes, we're talking about cameras, and our cameras determine our pixel size and our pixel distribution. So cameras only see in black and white. There is no color detection on a camera. They only see in grayscale, is what it's called. Now, grayscale images are quantitative. Grayscale images in bit depth are intrinsically linked. So if you go back to the first lecture and you remember the concept of contrast to set strikingly in appearance, in opposition of each other in appearance, you'll remember that we have different bit depths which modulate our contrast. Well, grayscale images are linked to contrast because the assignment of values generated by the camera are going to be assigned to a certain bit depth. We're going to get into that in more detail, but know that grayscale images are using mathematical values to generate the image. So the value of each pixel represents the electrical current that was generated by those photosensitive diodes on the camera detector chip. How this works is as a photon or a light hits that camera chip, the intensity of that photon is going to generate a different variation of electrical current. That electrical current is then transmitted to the computer and interpreted and assigned to a grayscale value. You can imagine a bright photon or a lot of photons is going to generate a stronger electrical current than a single photon or a dim low energy photon. And so we can get different distributions of grayscale values based on photon intensity or light intensity. Really bright lights are going to get strong currents. Dim lights are going to get really weak currents. And that also corresponds to the number of photons you have. More photons, bigger current. Less photons, smaller current. And so then each bit depth is going to correspond to a shade of gray in your image that is generated by the computer. And then the common convention is the less intense the photon, the closer to black it will appear. That can change post-processing. You can do what's called an inverted LUT, where you make dim photons be white and dark photons black, which can be useful, but we're not going to care about it too much for this. Just know the common convention is a dim photon is going to be closer to black. Okay, so the quantitative assignment of grayscale images. So we represent photons in a grayscale image, and we do that by using what's called the image histogram, which is over here on the right. First, I want to remind us of bit depths and what they mean. So a 1-bit camera is only going to see all black or all white. It is a 1 separation between black and white. A 2-bit camera starts to introduce shades of gray. Right? We have a black, a dark gray, a light gray, a white. 4-bit, so on and so forth. Well, on modern cameras, they go all the way up to 16-bit images. We don't always want to use the full bit depth of our image depending on what we're trying to do. If we want to introduce more contrast, we can lower the amount of bit depths that are used so that we get a more distinctive or more contrast separation between gray values that are being assigned to photons. But we care about generating a pretty picture and we don't care about contrast. So we're going to use the full 8-bit image. Now an 8-bit image is going to have 255 gray levels assigned to it. A 16-bit image is going to have something like 64,000 gray levels assigned to it. We're going to stick with the simpler math for now. So when you look at the image histogram, the first thing you're going to notice is that on the y-axis here we have a frequency. What that frequency is corresponding to is the number of photons, or the number of pixels, rather not photons, pixels that had a certain intensity assigned to them. This intensity measurement over here is a measure of the electrical current that was generated at the detector chip by the photosensitive diode. And you can imagine that this intensity can be modulated based on multiple factors. One being the energetics of the photon itself. A high energy photon is going to generate a stronger electrical current than a low energy photon. And then also the number of photons detected will correspond to a higher electrical current or stronger electrical current than a small number of photons detected will have a lower current generated, a weaker current generated. And so then these currents that are generated by the detection of photons are going to be assigned a bit depth of value. So we're going to say a really high or a really strong current is assigned to this 15 here on our 4-bit image, so it is completely white, and then a single low energy photon will be assigned to 1. So a weak current is going to be assigned to 1. So when you look at the image histogram, you're going to see binning of pixels with certain values associated from the current, and that's going to be called intensity. Right? So intensity is the culmination of currents being assigned to bit depth of values. This is kind of a hard concept to get across to people, so I'm trying to explain it in multiple ways. Our intensity is equal to the current generated and assigned to a bit depth of value. The current generated is determined by the intensity or number of photons that are detected by the photodiode in the camera chip. That is what makes these digital images quantitative, so that we can actually do analysis on a digital image, because the histogram was generated mathematically based on real data that was detected by the camera. I hope that makes sense. So when we look at this histogram, we can interpret this by we have a lot of pixels that fell into this kind of low intensity range, right? These are dim pixels. So this could be attributed to background. This could be attributed to a weak fluorophore. We don't really know exactly why without looking at the image, but we can attribute to this as dark pixels, and there was a lot of them. We can go over here to this next peak. We again get a lot of pixels that are assigned to this kind of intermediate mid-range intensity value, and we can interpret that as maybe that's a protein localized to a certain spot, maybe that's an aggregate of proteins that are not fully folded correctly, so we don't get the correct emission spectra of the protein. There can be a lot of things that drive that. And then we get this really big spike here. This is likely going to be the true data, right? This is going to be the true value of what you have, where you're getting a lot of pixels that have a similar intensity, a similar brightness. The word intensity and brightness can be used interchangeably in this case, where we have a lot of really bright pixels in this range. And then we get some hot pixels over here, which maybe that means we had a really strong localization of a lot of proteins. So we had, maybe we'll use easy to understand numbers, we had 100 proteins localized into a 1 micron spot. So those were all detected by the same photodiode, because remember our photodiode is 6.5 microns wide, and we had 100 proteins localized within a 1 micron spot. So all the photons from that one spot were collected by the same diode and generated a really strong electrical current, which made that thing really bright. Where over here, using that same thing, same idea, maybe we had the same 100 proteins, but they were distributed across a 12 micron area. So 2 photodiodes were recording the current from those 100 proteins. So we had a weaker current generated from the photodiode, which means it's a dimmer gray value. And so because of these relative brightness, relative intensities, we can then quantify the number of proteins, the brightness, the localization of our proteins in our image that we've generated. I hope this makes sense. Like I said, this is probably the hardest aspect to get your head around when it comes to quantitative microscopy. So if you don't quite understand this, let me know. I can go back through it again with you. All right, so now to interpret this digital image that we've created. We've made a grayscale image. So now we need some way to actually process and conduct our analysis. There's a lot of ways to do this. Basically, every company that makes a camera also makes an image processing software. I put 3 of the big ones up here, but your favorite choice is what you can use. The most commonly used one that you'll see in publications from Nature all the way through everything else is ImageJ. ImageJ is also Fiji. Fiji stands for Fiji is just ImageJ. It is a free open access software that is funded through the NIH. So it doesn't cost you any money to use it. And because everyone uses it, there's also a lot of programs and macros and scripts made for ImageJ to do the type of analysis that you want to do. The other one I threw up here is Amaris. It's a really cool 3D reconstruction software, but it costs you about $80,000 for the software. So do it in Fiji if you can, because that's expensive. And then Olympus makes what's called CellSense, which is what drives the microscope, but you can also do pretty much all the same functions that ImageJ can do, just using Olympus's software. So there's many software options available. Use the one that you like the best and never look back. So the first thing we need to do, right, is we need to be able to generate a real scale of the image you're looking at. Again, pixels are defined as the size of the photodiode. Cameras see things in pixels. So what the camera knows is how many pixels were used to identify an object. So if you have a bacterium that is 3 microns long, the camera doesn't read that as a 3 micron long bacterium. It reads it as a half a pixel long bacterium. Well, we don't report things in biology in terms of pixel size. We report things in terms of microns, centimeters, real distances that we can actually handle. So we need to be able to take that fixed distribution and fixed pixel size from our camera, and we need to convert that into a physical distance. So we want our bacterium to not be half a pixel in size. We want our bacterium to be 3 microns in size. How do we do that? So to convert to a real actual physical distance, we need to know the size of our pixel, which is determined by our camera architecture, and the magnification of our objective. Most modern softwares are going to do this for you, right? CellSense knows what camera you're using. They know what objective you're using. They just plug and play for you. But if we don't, we can use the relationship between pixel size and magnification size to convert to a pixel. We're going to do this the hard way to start with. But just know that most of the time you're just going to trust what the CellSense spits out to you because it's telling you it did the conversion correctly. So behold, the mighty micrometer. Right? This is a tool that we can use to calibrate our images in order to know what our real physical distances are. So on the left is a picture of a micrometer that I actually took. I know that the distance between each one of these dashed lines is 10 microns. And I know that my camera, I believe I said in the last slide, was 2,304 pixels by 2,304 pixels. So I know the distance between these 2 points, and then when I go into Fiji, it will tell me how many pixels are in between these 2 points, and I can match them up and I can say, okay, each pixel is equal to this many microns based on the objective I'm using. This is the hard way. Like I said, the easy way is just trust what CellSense is telling you. The software will most likely do it for you. But if you don't have that capability, you can put a micrometer on your microscope using the objective that you're going to take your images of your sample on, take a picture, then do your imaging. Then when you go to do your image processing, measure this distance in pixels. We know that this is 10 microns. And so then we're going to know that there's how many microns are in each pixel on the image that you're taking. So I want to leave you with one last example of how we can actually generate some real data based on experiments I did. So I showed you in the first PowerPoint, the first talk, the NLS import assay and how those import assays were able to tell us the dynamics of a protein. Well, here, this is the methodology I used to generate those import rates. So the first thing is I had an NLS sit with a fluorescent molecule on it. I also have some labeled membranes thrown in there. I have some MPC markers thrown in there. And I can merge all these things on top of each other to get one solid image. So I can use multiple fluorophores at once to see multiple things at once. But since they were taken at the same time, I can measure different things between channels because I have different wavelengths of light that are specific to a certain fluorophore. So this is just a little video I made of how I actually ran this in Fiji. So what you're seeing over here on the right, this is what Fiji looks like when it's opened. This is my image of a nucleus that has an NLS import signal in it. And I'm going to run through this video and kind of explain what's happening as it goes through it. Hopefully my video embedding works. Okay, so this first thing I've done is I've drawn this line through here, and I'm going to generate what's called a plot profile. So you can see I'm getting gray values assigned along that line. That's telling me the intensity values of all the pixels that lay underneath that. I then did a step that's called thresholding, and I'm using this thresholding based on the intensity values of the pixels to only isolate the pixels that fall under an intensity value I care about. So that's allowing me to specify that I only care about the proteins that were fluorescently labeled inside of this nucleus. I then run a macro, is what it's called. And what you can see on this slider here, I'm changing the intensity values along this slider to only include certain pixel values, right? Only certain intensity values of things. I then run this macro, and I can measure my pixels that were located inside of this region of interest. So I generate this region of interest around this nucleus, and I got some off-target ones. I simply deleted those. And then I get rid of my thresholding value. Now I have this region of interest that's going to measure the intensity value of every pixel inside of this region of interest. Well, that's not super informative to me. I want to be able to know other things, right? I want to be able to know what the value of this cytoplasmic protein is and the nuclear protein. So I just ran this little macro that made that ROI bigger and smaller. So I did an erosion and a dilation of the original region of interest. And then I spit out some data that looks like this, right? So it told me lots of different things. One, it told me the area of the nucleus I'm looking at of all those ROIs. It told me the mean pixel value of that intensity value inside of the nucleus. It told me the dimmest pixel, right, that minimum. It told me the brightest pixel and that maximum value. It told me what the perimeter of my region of interest was. This integrated density here, this corresponds to the image histogram we looked at earlier, where it's telling me the brightness associated of that protein of interest that's inside of there. So this is how we get from that quantitative histogram to real data. I'm measuring the sum of all the proteins inside of that nucleus and the brightness and the intensity of all those proteins that are located inside the nucleus. So that translates to an actual number of proteins. So I hope you're able to follow that. Feel free to rewind and watch it over again to see what I did. But the goal of this part of the step was to generate a region of interest so that I can measure the proteins inside the nucleus. And I'm going to go into that a little bit slower and easier to see on the next couple slides. So the first thing, right? So this is that thresholding step. What I've told the software to do is to include all pixels of a certain intensity in this image. And so I believe my cutoff was like 300, and my maximum, I basically made it full bore. So my background pixels here won't be included in the intensity profile. And then I can tell it to measure that thresholded image and generate what's called a region of interest around the nucleus. That region of interest will measure the intensity of every pixel inside of that region of interest. Well, because we know we had a fluorophore there, and that fluorophore generated an electrical current that was inside an intensity value, we can correlate the intensity of all the photons in here to an amount of proteins inside of the nucleus. We can't count like 1, 2, 3, 4, 5 proteins, but we can get the collective sum of all the proteins inside the nucleus. And then I was interested in also knowing what the amount of proteins that were available are outside of the nucleus. So that's where I do this dilation step, where I just take this ROI, I make it slightly bigger than the nucleus itself is. So this is now measuring the amount of signal, the intensity values I have in the cytoplasmic domain, plus all this internuclear signal as well. But I can remove these internuclear proteins by subtracting the value of this integrated intensity of the inner from this outer, which leaves me only the cytoplasmic protein. And then I can generate my eroded inner halo the exact same way, right? Boom. There we go. So there's my eroded inner halo, and I can compare my inner to my outer. There's reasons that we did that, which don't really matter for the sake of this video, but the point was just to demonstrate you can generate multiple types of ROIs based on thresholding of intensity values, which correlate to protein values. And then the really cool thing about this is I have generated these regions of interest around this rim of the nucleus here. Well, if I'm interested in the nuclear envelope, for example, I can transfer that ROI I generated based on the NLS localization inside the nucleus to another channel. And now I have an ROI that is specific to this nuclear envelope around the nucleus, and to measure the intensity of this nuclear envelope proteins, I can subtract this inner halo from this outer halo, and I'm left with the difference, which is what's in between them. And I talked earlier in my previous videos about the chemical dyes. This is an example of a chemical dye. It's just a dye that associates to lipid motifs. It's a lipophilic dye. So I'm labeling the entirety of the lipids along the nuclear envelope here, and then I can take the intensity value to know how much lipid is actually present at the nuclear envelope. So again, to kind of sum everything up and bring it full circle here. So we can apply this data. So using my halo ROI trick, I was able to measure the intensity of the proteins that were internuclear based on the intensity values generated from the ROI. I was then able to compare that nuclear intensity, which corresponds to proteins that have been localized inside the nucleus, to the cytoplasmic protein abundance, and generate this ratio of nuclear proteins to cytoplasmic proteins. That ratio then corresponds to a rate. So I have an import rate based on the intensity of proteins found inside the nucleus versus the proteins found outside the nucleus, which then gives us our rate, which we then were able to correlate to the binding affinities of certain proteins, which then gave us a mechanism for how different proteins get into the nucleus at different times. So I hope this was helpful. I know it got a little messy there. This is kind of a hard topic for people to get their head around, so I wanted to make sure I explained everything thoroughly. If you have any questions, you can always email me, and I hope you enjoyed.